present to the world what they've been working on, semi-in secret, for the last few years. Even some of us on the advisory committee have not seen everything, so it should be very exciting. I'm very excited too. I'm Kelsey's advisor, and Kelsey is my first master's student, so I'm particularly proud of this one, and I think she's setting a bar, to be honest. It's going to be very hard to follow. So I just am going to do a brief introduction, a little bit of her background, and kind of drum roll for her to take the stage and tell you all about her research. For those of you joining online, at the end, she'll present, I'll give an intro, then there'll be time for questions in the room. For those of you online, please type your questions in, and we'll be looking at those and bringing those up, challenging Kelsey on that. And then her committee and I will go and take her into a separate room to further, test her hypotheses. And there should be either white smoke or black smoke, but I've got a good feeling, and so then we'll come back and we can celebrate, hopefully, with her, depending on how things go. So I just have a few slides here I wanted to share, and how do we get this?
You may have to drag it over, because it's on project right now.
Where do I drag it?
To the right. There you go.
All right, so Kelsey Montalto, in her defense of her master's thesis. I can say just a couple words about Kelsey. Resilient, honest, diligent, unflappable. Okay, if you've seen her in the last week, she is just steady nerves. So, and she's also very bright and has done a wonderful job on her thesis. So, as I mentioned, Kelsey is my first student. She joined Moss Landing and CSUMB even before I arrived, and she was graciously co-advised by Scott. Scott, can you stand up for a second? Scott Hamilton, and it provided a wonderful, context and collegiality for Kelsey here. And as she was my first student, she was the only graduate student in my lab when she arrived. But we were soon joined by many others, and most of them are in this room today. But Kelsey didn't come, unprepared. Her love of the sea started at a very young age. And she had a great affinity always to animals. Now, I received a lot of photos and a lot of information from her wonderful family. Too much to go into. I could fill probably a whole hour, but a consistent theme was this contact with animals, and she just has this way, sort of a whisper. And combining those two, she became, more and more into the ocean environment and the animals that it contained, and making contact with them. But her specialty, dangerous animals. And I think this photo really sums up a lot of Kelsey's qualities. Like, see that clenched fist? Determination, enduring hardships with steadfast nerves, and her colleagues can only look on with awe, And so, but it wasn't always life on the sea, We all know she's an incredible baker, right? Incredible artist. But did we know that she had so many other talents? Let's just take a minute to unpack this here. She plays several instruments and was a band president, leader, line leader, also on a championship player in basketball. So these are parts of Kelsey that I didn't know about. Did you know about this? But it was really the science, okay? And she's very modest, but in her, attraction to the creepy crawly things that earned, did you know that she has an honorary degree also that, besides her undergraduate? Yes, yeah. She was conferred, I heard from her sister, that she was conferred the honorary title of Dr. Worm.
Yes.
You've never told us this. I don't know why.
It never came up.
That's wonderful. So this is not her first degree. And so she did graduate from Fresno also, so her other degree. And that's when she came and joined us to study the big dangerous fish, baby white sharks out here in our bay. And we are the better for it. She was, like I said, the first of a number of students now, and has provided wonderful mentorship in my lab and in Scott's lab, not only with the other graduate students, but with the undergrads. And she's continued to come to my undergraduate classes and help them, and she's actually earned the title of Shark Wrangler also here. So she's always at the center, always handling the animals. She loves to be there. She's never missed it. She's never not shown up, ever, throughout her career. So those are just a few wonderful qualities. She also has a fun side through all that seriousness, you know. And she's had wonderful experiences and friends here at Moss Landing and CSUMB. And while she was here, she racked up a huge number of accolades. Two-time recipient of the WAVE Award, the Jim and Peggy Award, Myers Grant, Coast Field Experience
Twice.
Award. Pardon me?
Twice.
Twice. Two times two, okay? Goes on and on. I didn't really have enough room in this slide for everything. She's a shark ecology and otter scholarship. She TA'd for my quantitative methods in marine science, like three times? 2 times?
3 times.
Amazing. And now she's a finalist on a Sea Grant Fellowship. Fingers crossed. The list goes on. And how many graduate students can say they've already talked, you know, on CNN about their project? So that is also a wonderful accolade for Kelsey. So she's going to give her talk. We're going to sit back. I'm very confident. She's done a lot of heavy lifting quantitatively, and will take us on a journey to talk about her thesis. Please welcome Kelsey.
Let it be known that a lot of those photos I told my family to send in. Yeah.
We weren't quite sure how much we needed to do.
Yeah. I let them know just how much, don't worry. All right, well, without further ado, I'm glad I was able to trick you all into coming today to talk about sharks when most of this is going to be about oceanography. But regardless, we are going to be talking about juvenile white sharks in Monterey Bay today and the effects of thermal habitat patchiness on local densities of a novel range-shifting predator. So essentially, we're going to be looking at why newly established juveniles in the Monterey Bay are selecting particular areas here. We know that they've moved here because of climate change, but the smaller spatial scale is a little more fuzzy there. And this is important because their selection of certain sites could impact the ecosystem throughout perpetuity as they are a new predator here. So before we get into the nitty-gritty, here's a bit of an outline for you. We're going to start with the introduction, then move into the methods, which consist of remote sensing and tagging. Then we're going to get into the results, discussion, and finally acknowledgments. And so if you ever get lost during this, all of those sections are up at the top in those arrows, or if you want to know where we are, and depending on what section we are in, it will be highlighted a darker blue. All right, so let's get into it. Climate change, we know, has affected species ranges and has been shifting them globally. For marine taxa, this typically means that they shift into poleward waters or into deeper depths. And this is because as water is warm, the coolest water that they can access is either further towards the poles or in deeper water. So on average, poleward, we've seen about a 6 kilometer per year shift for marine taxa overall. And for demersal fish, fish that live towards the bottom of the water column, on the sea floor, we've seen a 4 meter shift deeper in the North Sea. And so you can see that on the figure on the right-hand side here. These 3 different species of marine taxa here in the North Sea moving from their southern portion all the way up to this new northern range. These changes can be gradual and long-term. For example, this has been seen in the expansion of tropical herbivorous fish that come into previously too temperate kelp forests. Like you can see in the top left, they eat them down because they have all of that available food. It turns into a barren, and then that can have lasting ecosystem changes, where this one in particular in Japan turned into a coral reef system. Or these shifts can be abrupt after heat waves or El Nino Southern Oscillation, or ENSO events, occurring about on a 3 to 7 year cycle. And this has been observed off of the coast of California for 37 species between just 2014 to 2016. So you can see from the map on the right-hand side, we have 3 of those species specifically from top to bottom. That's a nudibranch, a barnacle, and then a sea butterfly. This was their northern limit prior to 2014, 2016. And then following that heat wave, they moved all the way up there. So that's a pretty big trek for those little guys. And so the variability of length and intensity of these rain shifts is highly influenced by how these organisms are interacting with the new properties of their environment. So you can see by going to an environment like the picture on the right-hand side, there's a lot going on. There's a seamount further out, there's a kelp forest, then there's the intertidal zone, and whether these organisms can survive, whether they can even get there, whether they choose preferred sites, is all dependent on whether they can have food availability, whether there are predators there, whether the physical environment is suitable to them, if there are too stressful of currents, temperature, potentially there's a flume of runoff coming from a river that's really turbid or has chemicals that's harmful for them that they don't want to be in. All of those can impact whether they continue in these areas. And survivorship in these novel conditions can lead to the ability for consecutive range expansions. For example, if there was a suitable habitat a little bit north and they're able to get there, then potentially that could repeat further and further north as they go, serving as habitat stepping stones for them. And this was observed in Kellett's whelk that was predominantly just in Southern California historically, and then through heat waves they were able to establish new source populations moving north and north through great recruitment years and then establishing a steady population. So bringing that to our system here, we have the California Current System, which runs from Canada to Baja California, and it has a very strong thermal gradient. So you can see from this figure here, which shows the climatological mean, or in other words, the long-term average of temperature for the area, which makes that nice, smooth temperature gradient, where we have the deep, dark, purpley blue towards the north, and then that bright, hot red down south. And so it has a really smooth gradient here because this is the climatological long-term mean. And then if you zoom in temporally through time, it does vary seasonally. So in the winter, we see some variation, and this is associated with the Davidson current. In the spring, we see high upwelling, and in the summer, we see a relaxation of this upwelling. And then if we zoom in even further to a single day, like you can see from this figure on the right-hand side, this is a single day of sea surface temperature. It's a lot more patchy and dynamic as the California Current System interacts with a complex coastal boundary. So you can see this manifested thermally through that figure, where we have patches of those deep dark blues and purples contrasted with bright greens and yellows and oranges. So it's not that smooth that we saw from the climatological mean. Now, looking at a single day, it's really, really dynamic. And so I mentioned this previously, but just for clarification, upwelling is one of these oceanographic processes where winds parallel to the shore, here those would be northwesterly winds, with the Earth's rotation will pull surface waters away from the coast, leaving a deficit that is then filled by deep, cool, nutrient-rich water. And so as specifically this oceanographic process interacts with the complex coastal boundary, we get another phenomena, which is the upwelling shadow, which is a warm surface patch compared to the rest of the area that persists for an amount of time that is the result of local topography and local and regional wind forcing. And so we actually have one here in the Monterey Bay. It's in the northern portion in Soquel Cove, called the Soquel Cove upwelling shadow. And you can see how this interaction actually works from the diagram on the right-hand side, where the upwelling jet that comes off of Point Año Nuevo at the top comes down. That's that big bold arrow that comes in, and then it hits the point, but then the embayment eddies in on itself similarly to eddies on the edge of a river that are much slower moving than the rapids towards the center. And here we have much more stagnant warm water. And that's physically manifested in sea surface temperature, where we can see this hot spot, this really warm spot in the northern portion of the bay right within Soquel Cove due to this upwelling shadow. And so bringing this to the model organism that we've been waiting to hear about, white sharks. The white shark is a lamnid shark, part of the family Lamnidae. It is a highly mobile predator. It does have a global range, which you can see on the right-hand side, but this is way bigger than the study system I just introduced you to, and that's because this is the adult range. They do experience an ontogenetic shift throughout their lifetime, which essentially means that when they are juveniles, they eat and inhabit different areas than when they are adults. As I said, this is the adult range, not necessarily the juveniles that tend to stick closer to the coast. And part of this is because they are endothermic. Like you and me, they can generate and maintain their own body temperatures of about up to 25 degrees Celsius, which you can see from those really bright red internal core temperatures there. And so how this manifests in its ontogenic shift is also in their diet. So adults predominantly eat marine mammals, and they have the much wider, triangular, serrated teeth that are used for chopping up these large animals to swallow them and eat them. And the juveniles, on the other hand, eat fish and small elasmobranchs, where they use those really pointed, more conical teeth to puncture and then swallow whole. In addition, there's a difference in the coldest temperatures they like to be in. For adults, this gets as low as around 9 degrees Celsius, and for juveniles, it's much higher at around 15.1. They just don't have as much body mass to maintain their thermal inertia compared to the adults, and this is also seen in their size. So for the adults, they can get up to 6 meters long, which is really, really big, and for the juveniles, their size is around 1.5 meters when they're young of the year. And this is also seen in their life cycle. So here on the right-hand side, we have their historical range through their life cycle, where the smallest sharks represent the juveniles down south of Point Conception there, where they're sticking close to the coast in their warm waters. And then as they grow larger and larger, they're able to tolerate colder waters, moving further and further north until they are large enough to go offshore as adults and then come back, pup, and start the cycle all over again. And what's really interesting about this is that a range shift for juveniles was observed on a regional scale, where the juveniles, after the 2014 heat wave off of the coast of California, known as the blob, moved from their northern range limit, which is depicted here at Point Conception, all the way up to Monterey Bay. And so not only were they novelly seen here for the first time after 2014, but we also saw an increase of these juveniles, and especially within Soquel Cove. So this figure on the right-hand side, each panel is representing a different year for the bay, and each of those orange circles is representing an observation of a shark, going from one observation in 2014 all the way to 19 in 2019, where you can see not only is the number increasing, but they seem to be aggregating in the northern portion of the bay where that Soquel Cove upwelling shadow is. And it should be noted that in addition to the size differences that help distinguish adults from juveniles, we also see the juveniles during a different portion of the year. So adults are pretty frequent to this area, but typically during the winter months. And then the juveniles we see during the spring and summer. So that helps us distinguish between them as well. And so there's an increasing body of work on these juveniles, but nothing really on the smaller spatial scale, which brings me to my research question, which is what environmental variables are associated with juvenile white sharks within Monterey Bay? And so I chose to address this by using two different techniques, the first being remote sensing, where I was assessing their surface behavior, and I was interested in looking at variables such as sea surface temperature, chlorophyll, and turbidity. And then the second technique was tagging, where I was looking at depth behavior, and I was using in situ temperature, or a temperature collected directly in the field, sea surface temperature, chlorophyll, and turbidity. And we've got these two icons here, the drone on top next to remote sensing and the tagged white shark down below. You will see those appear in the top right-hand corner, depending on which methodology and results I'm talking about throughout the rest of the talk. So if you get a little confused, because it might get a little confusing, those are there for your reference. And then to break it down a little more, here are my hypotheses. So for my first research question in regards to the surface behavior for remote sensing, my first hypothesis was that juvenile white shark numbers will increase with sea surface temperature. In other words, since they have had previous research that shows that they're associated with temperatures and they're trying to thermoregulate, it would follow that they are looking for warm waters. And then including sea surface temperature spatial anomalies will improve predictability. I'll get into what that metric means, but essentially what it's trying to say here is that where we see them, whether it's warmer or colder than the rest of the bay, will help improve where we see them, because if they are seeking warm waters, this spatial differentiation will help us find them. The inclusion of chlorophyll will improve predictability. Chlorophyll is a proxy for phytoplankton biomass, and so if there's more phytoplankton, there could be a bottom-up effect where that could lead to more prey, which then could sustain a larger concentration of juveniles. And then finally for this portion, juvenile white shark numbers will decrease with turbidity. Turbidity here being a measure of water quality, where the thought behind this is that as they enter waters that could be more turbid, it could impede their ability to hunt if they're relying predominantly on sight, or that it could be too silty for them and it might gum up their gills compared to Southern California. For the tagging portion, I only had two hypotheses. The first being that acoustic detections will increase with temperature at depth. This follows the same reasoning as the remote sensing. Warmer waters would be associated with more sharks as they are trying to warm themselves. And then that the inclusion of chlorophyll and or turbidity would improve predictability of juvenile white sharks. And this is essentially the idea that including more environmental variables will help us understand what is driving them to select these sites. So having a better holistic idea will provide a better answer. Okay, so getting into the methodology, starting with the remote sensing portion, drone surveys were conducted to assess juvenile white shark abundance from 2017 to 2024, mostly in the months of March through October, at two sites, being Aptos and Marina, which are depicted on the map here, with the northern one being Aptos, right within that Soquel Cove upwelling shadow, and the southern one being Marina, which is in a little bit of a more exposed portion outside of that upwelling shadow. And so this is what a drone survey would look like. Try your best to see how many you can count. Put you in the shoes of one of our field samplers. And so our drone pilot would go and conduct surveys where they would run a lawnmower pattern parallel to the coast, looking for sharks as they went along and taking video that they would reference later so that they could double check that they weren't counting kelp. Because I'm sure you can tell from this video that it is a little deceiving here and there, especially with a little bit of turbidity coming in and out and a little bit of the kelp rack coming in and out. So the silhouettes can be a little hard to distinguish when you're only doing one pass. And if you guessed 5, you were correct. There were 5 in this clip. And then moving on to the satellite data, these were used in tandem with the drone survey counts. And I used 3 environmental variables here. I used sea surface temperature, which you can see from the top hand figure there, which I collected from the MER data set, downloaded from there. And then the other 2 that I used were chlorophyll and turbidity, which I sourced from the Aquamodus data set, which are the left and right bottom photos, respectively. So again, chlorophyll A being a proxy for phytoplankton biomass, and turbidity being a measure of water quality, where here in the glasses, the one on the left is not turbid water at all, it's very clear, and then the one on the right-hand side is very, very turbid, silty water. And so all of these environmental variables were downloaded as daily values for the same length of time as the drone surveys were conducted, from 2017 to 2024, and within a bounding box of Point Sur to Pescadero, so not just the Bay Area, but the surrounding areas as well, so that we could look at some interactions on a spatial scale that included the greater area so that the site selection could be a little more definitive. And then these were masked to include depths of less than 1 kilometer deep, because previous research has documented that juveniles tend to inhabit coastal waters that are only up to 1 kilometer deep. And so this would look like what is shown here, where the picture on the left-hand side is sea surface temperature for 1 day for the entire bounding box. And then after applying that bathymetry mask, you would get something more like what's seen on the right. From there, I didn't want to use just the raw values. I was interested in some spatial comparisons and temporal comparisons, so I calculated a bunch of anomalies. Anomalies here being that I subtracted a set value from the raw values to get a metric of whether it was above or below that set value. And so, for example, the first set of anomalies that I calculated were the spatial. So if you would imagine that this stack of images here was my entire data set, I would take a single day of that. I would then average it to get a single value, subtract that from the raw, and then would get an outcome sort of like this, where you can see the color pattern is the same, because I'm subtracting a constant, but now it's centered around a 0, so the color is a little bit different. And then in addition to those spatial components, I was looking at temporal. So I did a bunch of climatological anomalies. I did 4 of them. I did a long-term, annual, seasonal, and monthly. And so instead of subtracting the mean for the entire bounding box, what I did instead was subtracted means for a single pixel throughout whatever time period that was. So for the long term, I would take the mean value of that pixel throughout the entire data set and subtract that mean from it every single day. And for the annual, within a single year, seasonal within a season, and for the monthly, within a month. And then in addition to that, I did a physiological anomaly, but just for sea surface temperature, where this figure over here is a plot of the experienced temperatures of juveniles within their historical range in Southern California, where they're just swimming around within the water column. And so I took the lower bound of the 95% confidence interval at around 15.1 and said this is typically the coldest that they are observed to be found in. So if I subtract this from every sea surface temperature pixel, this will give me an idea of whether they could be physiologically selecting. From there, I did site-specific pixel selection, where I selected the pixels that were within 2 kilometers of those drone survey sites, Aptos and Marina, and then averaged them to get a single value per day. So you can see that by the site selection, I would end up with clusters and then average them for a single day to get 1 value for Marina and 1 value for Aptos. And from there, I could move into my data analysis, where I ran generalized additive mixed models. I know that sounds a bit wordy, it sounds a little bit fancy, but essentially what these were doing were I was combining and adding in different combinations of variables to see how they interacted with each other to predict juveniles the best, and also their relative impact on the juveniles. So in other words, say that you were me. I run very cold. I like warm weather, if that's a reference for anybody who doesn't know me. So say I was trying to move somewhere. If I was trying to move somewhere new, I'd probably want it to be warm, right, so that I wouldn't be cold. I would want it to be close to grocery stores, so access to food. I would want it to be clean, not have a bunch of air pollution, and maybe I'm looking for a house instead of an apartment. So these are kind of the analogous compromises for the sea surface temperature, chlorophyll, turbidity, and site metrics that I used within these analyses to predict abundance of juveniles. And then for the tagging portion, I deployed 4 moorings off of the coast of Capitola, New Brighton, Seacliff, and Aptos Creek. Each one of these had a VR2W acoustic receiver and temperature sensors. These were deployed at about 30 feet of depth and about 1 kilometer apart, so that the radius of their reception for the tagged sharks would not overlap. And then once they were deployed, we had to go out and tag them. So the video on the bottom is of me waiting to get in proper, perfect position for a shark that is a little too far away, a little too deep. The water here is also a bit turbid. But we went out into Soquel Cove in late June and early July and tagged 7 sharks with the tags that you can see on the top there that have a titanium dart that goes into their skin, and then the thicker cylindrical portion there of black is the actual transmitter that is detected by the receivers. And then from there, I could use those detections to get metrics of abundance and residence time. So we let those stay out for a while, and then we had to go and get them. So we used 2 techniques for retrieval in late October and early November, the first being scuba. So you can see from the image on the left-hand side, that is one of my scuba crew that were so willing to go out with me, even though the waters looked as green and gross as you can see in that video on the right-hand side where I'm trying to get that receiver off of the mooring. And then the other technique that we used was grappling hook. So we went out on the boat and we had 2 grappling hooks attached by a weighted line, and you can see the picture of me on the left-hand side with that grappling hook ready to deploy it, and we did our best to ensnare them, circling around with the boat, and then we would try to get them as close up and then take the anchoring system off and get it on board, which you can see from the image on the right-hand side. And you can tell who has the most investment in this project by what level they are on the boat. So you have me, belly down, really making sure that we've got it secure, Sal kneeling, and then Duncan's, there's an extra set of hands. And then once we had those receivers back, I was able to download all of those data. And from there, I calculated metrics of the total number of sharks per day and also the total number of detections per day, which I used as a metric of residence time. Any day without detections was considered a 0. So you can see from the abacus plot on the left-hand side, these are the detections for the 7 sharks, which are the different lines on the y-axis here, throughout the time of deployment. So each of those black dots is when a shark is showing up. So anywhere where that really light gray line doesn't have a black dot, that's a 0. And then I also downloaded all of the in situ temperature data and averaged them to get a single value per day because they were sampling cyclically throughout the day. And then I combined these by site and date. From there, I wanted to use as much data as possible in my analyses, so I combined these acoustic data with the satellite anomalies and raw values, and followed the same pixel selection process as in the remote sensing portion, but here used a process where I selected them within 1 kilometer of site instead of 2. And I combined them all by date and site. And then we were interested in whether time of day would have a different effect on these sharks. And since the acoustic sensors were detecting multiple times throughout the day, we could look and see, take the original combined data of the total number of sharks per day, residence time per day, and the mean in situ temperature, and look at it through hours to see if it had a certain time of day effect. So they were grouped by hour, and means were calculated from there. And then I was able to get into my analysis. So for the combined acoustic and remote sensing data, not the time of day, I ran GAMS again, and this time including the variables of in situ temperature, chlorophyll, turbidity, and site for both response variables. You may notice something is a little different than the last section. Here we only have in situ temperature, and the reason that only in situ temperature is here is because the models with just the field temperatures performed better than in combination with sea surface temperature or than just sea surface temperature alone. And then for the time of day acoustic data, I ran GAMS including in situ temperature, hour of day, and site for both response variables. Okay, so now we've gone through all of the background and the methods. Let's get to the meat and potatoes of the results. So for the remote sensing, here also to orient you at the bottom, the model formula will be listed. If you're really interested in it, you do not need to know, and you can ask me about it later because I probably ran over 150 models total. But the best remote sensing model showed that more sharks are seen at Aptos than Marina. And so you can see this on the right-hand figure where this is split up by those different seasons, where those red boxes are higher than the blue ones, except for in the winter, and that's typically probably a result of the fact that we don't do as much drone sampling in the winter and that it might be too cold for them here. And this was a significant relationship here. Okay, this plot might seem a little scary. Let's orient you to it. So on the bottom, we have our variable that we're interested in here, which is absolute sea surface temperature, and then on the y-axis is a partial effect. What does that mean? Well, to give you a little bit of, this is how the GAM actually outputs it, but all you really need to know is that 0 line means that there's no effect. It's not really positively or negatively influencing those sharks, but anything above is a positive influence where you should see more sharks, and anything below would mean fewer sharks. So if you get lost in the orientation of this graph at any point in time, follow the sharks, follow the arrows, follow the more, follow the fewer. And then every tick along the x-axis represents a sample, and then that thick black line is the actual trend for this variable. So for absolute sea surface temperature, we see an increase in juvenile white sharks when waters are warmer, from around 13 to 17 degrees Celsius. And this is a significant relationship. And what's really interesting about this is that that gray shaded area is the confidence interval, and we see it above that 0 line from that narrow band there from about 13 to 15 degrees Celsius. And this is really interesting, but we'll come back to that in the discussion to talk about it a little more because I want to get through all of the results. So going back to my hypotheses, for juvenile white shark numbers increasing with sea surface temperature, there's a little bit of a mixed bag, yes and no. We saw that there was an increase from temperatures of 13.2 to 17.5, but about a neutral effect after that and a negative effect before that. Now we have an even more complicated model output here. This is an interaction between two variables within the model, which are turbidity spatial anomaly on the x-axis, which essentially just means that it is either more turbid or less turbid than the rest of the bay. And then on the y-axis, we have the sea surface temperature spatial anomaly, where it means that anything above 0 is it's warmer than the rest of the bay, and anything below is that it is cooler than the rest of the bay. So for example, a way to read this is that we, in the bottom left-hand corner, we see it's kind of that 0 white color. So when it's less turbid than the rest of the bay and it's much cooler than the rest of the bay, there's not really an effect on the sharks. We're not seeing more of them, we're not seeing fewer. But where we do see the most juvenile white sharks is when the sea surface temperature is warmer than the rest of the bay. And so we consistently see this really red color up when it's at least a degree warmer in these sites than the greater bounding area. And this is in contrast to when it's much cooler down at the bottom where it's that deep blue, and especially when you see that waters are way more turbid than the rest in the bottom right-hand corner where it is especially blue. However, we do see this really interesting effect when turbidity is slightly elevated, and the spatial anomaly is negative. So when it's cooler than the rest of the bay, but a little bit more turbid, not super turbid, but a little bit. And this is a significant relationship. And so we'll get back into why this might be a little bit later on, but it's quite interesting. So going back to my hypotheses, including the sea surface temperature spatial anomalies will improve predictability? Absolutely yes. The top model did include the sea surface temperature spatial anomaly. The inclusion of chlorophyll will improve predictability? Yes, it did, but I didn't talk about it because turbidity did way more. Through the model selection process, including chlorophyll did improve the model's predictability ability, but turbidity was consistently better, so we're not talking about that in the results today. And then juvenile white sharks will decrease with turbidity. Again, this is a little bit of a mixed bag, yes and no. We see a positive effect observed when these levels are slightly elevated, but otherwise we see a fairly negative effect on the juveniles. And then getting into the tagging of it all, the time of day acoustic model showed that there was a difference between site, where the most sharks were seen specifically at Seacliff, and the highest residence times were seen off of Seacliff here. And these were significant. And then the top acoustic model overall showed that we see more sharks when temperatures are cooler than around 14.5 degrees Celsius, which is an opposite trend of the surface behavior, and we'll dig into why that might be a little bit later, but we can see that it's positive before 14.5, and then after that we get really negative. And the same is true for the residence times, where we see a positive effect below around 14.1 degrees Celsius, but a negative above. And it should be noted here that the confidence intervals are pretty big, and there is a lack of sampling at the lower end of the in situ temperature, so an increase in sampling could potentially impact this trend. And again, this was a significant relationship. And then getting into the time of day here, there are a couple more components that you can see here where it might be a little tricky to interpret, but I've added in where the average sunrise and sunset times are at 6 a.m. sunrise and 8 p.m. sunset. And then the daylight hours have that sun icon at the top, and without daylight have the moon. And so we actually see higher residence times between 9 p.m. and 7 a.m., which is not what we thought, because if they are trying to thermoregulate, you would expect them to be at these areas when the sun is out, when it's warm at the surface. But something else clearly is going on here, and this was a significant relationship again. So we'll also dig into that in a little bit. This plot here is showing you a difference in residence times for the site depending on whether these sites are warmer than the rest of the bay or colder. So at the top, it's when sites are warmer than the rest of the bay, and at the bottom, it's when sites are colder than the rest of the bay. And then the size here indicates the amount of residence time, and the color is just site. So here we see that there is higher shark residence time when sites are warmer than the rest of the bay. We see larger dots in that top panel. And then in addition, we see no sharks observed at some sites when they are cooler. So we only see them at the Seacliff and Aptos sites in the bottom. And this was a significant relationship again. And so what this means, again, we'll dig into in the discussion. So going back to my hypotheses, acoustic detections will increase with temperature at depth. And here we actually see the opposite. So no, white shark abundance and residence time decrease when at depth temperatures are around 14 degrees Celsius, showing an exact opposite trend to those of the remote sensing data. And then these models also show that there are more sharks when turbidity is above the average. So this variable here is the turbidity monthly climatological anomaly. In other words, what this means is that the waters are more or less turbid than that month on average. So here we see when the waters are more turbid than that month typically is, we see an increase in juveniles. However, again, the confidence intervals here are very, very wide and don't really go above 0 at any point. So even though this is significant, it is only marginally so, and it should be taken with a grain of salt. And then going back to my hypotheses, the inclusion of chlorophyll and or turbidity will improve model predictability. And here, yes, turbidity was a key component of all of the top models. And chlorophyll did improve the models as well similarly, but again, not as much as turbidity. And so what does this all mean? Trends in sea surface temperature were seen to be within those documented in Southern California. So you can see again from the bottom panel here, that was the temperatures that juveniles swimming within their historical range are observed in, what they're swimming through, what they potentially prefer. And then once you plot that next to the available sea surface temperature just at the surface within Monterey Bay throughout the entire duration of my study, it's colder, which makes sense because we are in a more temperate system here than down in Southern California. Interestingly enough, the overlap between the two here is almost exactly where we see that narrow band above the zero partial effect from about 13 to 15 degrees Celsius. So although it's slightly cooler and Monterey Bay is a more temperate system, they are still within the bounds of their thermal envelope depicted in Southern California. Additionally, the site preferences or the difference in sharks during the different seasons, so the most in spring and summer, is likely tied to the fact that the sea surface temperature spatial anomaly is suitable for them. The waters are warmer than the rest of the bay, which you can see in the panels on the bottom. However, we do see fewer when sea surface temperature is above 17 degrees Celsius, and this is likely because at this point in time, the sea surface temperature is warm throughout in perpetuity. It's or rather ubiquitously, and they're no longer limited spatially to just the areas where we are conducting the surveys. They can go offshore. And then in addition, we see the fewest sharks in the winter, and this is likely correlated with the partial effect where we see fewer when the sea surface temperature is below 13 degrees Celsius, and this is probably just because it's too cold for them. It's too cold. It's really cold at our sites in that deep blue that you can see in the winter panel, and they're probably someplace warmer, staying nice and warm. Additionally, the inclusion of both sea surface temperature and the sea surface temperature spatial anomaly within the top remote sensing model shows that they are selecting sites when they are warmer than their surroundings, when the sea surface temperature anomaly is above 0, and then also when temperatures, raw sea surface temperatures, are within their thermal envelope for this area. And then we see the highest concentrations of the juveniles when these two variables overlap. So for example, you can see from the figure on the right that not only are the waters in the upwelling shadow really hot at around 16 to 17 degrees Celsius, but compared to the rest of the bay, which is around 12, 13, 14 degrees Celsius, it's much warmer. Additionally, the fact that there were more sharks seen at the Aptos site than Marina, which you can see on the right-hand side, suggests that Soquel Cove could serve as a thermal recovery habitat. Again, the Aptos site is within that upwelling shadow, whereas Marina is not. So the warm waters are more consistent there, which means that they know they can go there and warm up if they're moving offshore or deeper to exhibit other behavior. And this behavior is seen in other pelagic predators like blue sharks and molas, so it would follow that the same could happen for the juveniles. Additionally, when looking back on these two plots here, these two panels, where it's either warmer than the rest of the bay or colder than the rest of the bay for residence time, we see that sharks are selecting these areas for thermoregulation, it seems. They have higher residence time when the sea surface temperature spatial anomaly is positive, when it's warmer than the rest of the bay. And then, in addition, we see that some sites are more suitable than others. So when these sites are cooler than the rest of the bay, we only see them showing up at 2 of these sites. And you can even see it on both panels where the sizes of those circles are not the same. There are preferences between them. It should be noted that trends in sea surface temperature are related likely only to surface-specific behaviors. Thermoregulation specifically, and drone survey sampling only represents part of that behavior. So if you were a shark and you were trying to warm up, out of these 2 panels, where would you go? You would, for the top one, for the depth profile, you would want to go to the surface where it's that red warm, and then for the spatial anomaly map, you would want to go into the northern portion of the bay. But the drone surveys only capture them when they're at the surface, so we can only really see them there, which means that there's a specific behavior being exhibited here. Additionally, I mentioned that the tagging portion of my results showed an opposite trend to that of the remote sensing, where instead of the positive peak at around 14 degrees Celsius, we actually see that 14 is the maximum where the positive effect happens, again with those plots on the right-hand side with shark abundance on the top, and residence time on the bottom, where we see a negative effect after you reach around 14 degrees Celsius. And this suggests that something else is happening here. They're not thermoregulating. Instead, they're probably foraging. They're likely using the warm coastal surface water to regulate during the day, where we have those drone surveys, and then they're using the cooler, deeper, slightly offshore sites where these moorings are to hunt. And this has been observed in Southern California, where this is a plot of the tagged white sharks down there, their average depth per hour. And so you can see that they consistently go through patterns of warming at the surface, where it's kind of that peachy red color, and then they'll dive down to depth, where it's that darker blue and green, and then continue that process coming up to warm themselves. Additionally, this is supported by the higher residence times near these moorings during the non-daylight hours. Again, if they were thermoregulating, we would expect them to be there when it's sunny and the surface waters are warm. But instead, we see the increases when it's non-daylight hours. And so similar trends have also been observed in Southern California, so we're pretty sure that this is what's happening. Additionally, when talking about turbidity, we're back to the complex graph here. Again, focusing on where it's red and bright and we see the increase of sharks the most. we are seeing the brightest reds when that turbidity spatial anomaly is a little bit high but not too extreme. So when it's slightly elevated, and there's an intermediate kind of Goldilocks zone when they don't like it when it's too clear, and they don't like it when it's really turbid. They like it when it's just right. And so what's probably happening here is that since turbidity was a key variable in the top models, it could be that more suspended particles in the water means that there's more solar absorption, which means there's more heat. So you can see from the figure on the right-hand side, each of those columns representing a water column, the left one being very turbid, the temperature from that light source being the sun, making it really warm, and then getting all the way to the clear on the right side where the temperature would be a lot cooler as it penetrates all the way through the column. And this has been documented in surface waters coastally, where you would find a highly stratified mixed layer that's really warm at the surface and then much cooler waters down below. Additionally, it's possible that more turbid waters could aid in predatory behavior, where they could be using other keen senses instead of just their sight. So white sharks have a lateral line that can help detect changes in water pressure. They also have their skin for touch and temperature. They have their hearing, their sight. They have their mouth and nose for taste and smell. And then distinctly, they have what's called ampullae of Lorenzini. And so these are gel-filled pores, which are pictured down below, which are those little black dots that can help with electroreception, which not all marine taxa have. Distinctly, elasmobranchs have them. And so they could be using the turbidity to camouflage in with the rest of the area and be using those ampullae of Lorenzini to give them an advantage while hunting, which has also been documented in lemon sharks, so it follows that the juveniles could be exhibiting this behavior as well. On the other side, high turbidity could deter sharks, so that negative side of that Goldilocks zone. So this could be as a result of harmful algal blooms, which have been known to negatively impact ecosystems, and if any part of that has a phytoplankton type that can produce domoic acid, that could also be negatively impeding them. And so you can see from the figure on the right-hand side that harmful algal blooms create hypoxic and acidic conditions, where the top panel A is a nice and healthy ecosystem where the sunlight can penetrate, there's a lot of mixing, the fish are happy, the waters are oxygenated, but then you move to an ecosystem where there is a harmful algal bloom, all the sun is staying at the surface in that mat of phytoplankton, the water is highly stratified, there's not a lot of oxygen down there, and the fish are either very unhappy or they are dying. In addition, domoic acid has been documented to poison larger animals like the sea lion you can see on the left-hand side, so it is possible that it could be irritating or poisoning the juveniles as well. So it could be impacting the prey of the juveniles through the harmful algal bloom, or if there's domoic acid present, it could be irritating or poisoning them. So to start to wrap up, here's a little bit of a technique comparison for you, where the remote sensing technique was used to document their surface behavior. It allowed me to use long-term open source data of multiple variables to investigate a large spatial range, which helped indicate distinct trends by utilizing such a large data set. However, it should be noted that use of such a large data set could have underestimated p-values by finding subtle trends significant or through autocorrelation because there was a spatial component to all of the data I was using. And then tagging, on the other hand, was used to assess at depth and diel behavior and could assess a finer spatial scale. It could additionally record sharks anywhere within the water column, which was a distinct advantage over the remote sensing, where we could only record them when they were close to the surface. And it could collect data continuously, which allowed for a finer temporal investigation for that time of day to find those distinct trends there. So overall, this research added new insights. It added a better understanding of fine-scale selection for a range-shifting predator. It showed associations with turbidity and chlorophyll, which is more than has been previously done. It showed the influence of the sea surface temperature spatial anomaly, which to our knowledge is a novel parameter that's used for this. And it also showed the importance of complementary sampling techniques. By using both the tagging and the remote sensing, we could get a better idea of this difference in behavior, but if I had only used one, there's no telling that I would be able to see this trend as well. And so all of this research together could be used to predict stepping stones and model projections in the future, not just for white sharks, but this is applicable to other organisms and other ecosystems. And this could potentially help inform management efforts where, for example, right now, there are restoration efforts to help reintroduce otters to their historical range within the bay, but the presence of knowing when and where these white sharks are showing up could deter the restoration efforts in general. And it could also help to inform the public. It's always great to keep them involved and to know so that they know when the sharks are here, whether that means that they adjust their behavior to help them or also to help reduce the anthropogenic effects through potentially hitting them with boats or training them to be around people in ways that they are not used to. And so with that, we're now in the acknowledgement section in the final stretch. I want to thank my committee, or should I say my committee. To Sal, thank you so much for providing me the opportunity to come here. Moss Landing was the only grad school that I applied to within my cycle, and I had no idea whether anything was going to stick. And then halfway through waiting to hear, I got an email from you, and you weren't even faculty yet. So you took a chance on me, and we jumped into this pool together, and you've challenged me every step of the way, and I definitely wouldn't be as strong of a scientist or person in general if I had not been put through the steps that I had been under your advisory. To Scott, you have challenged me in other ways. But you have been in my corner throughout all of this. I don't know if that's because we have that Fresno bond, or yeah, go dogs, or if it's just because you took me under your wing as I was coming through, and Sal and I were both learning what we were doing. Whether it's been helping me out and challenging me through classes, or in my research, or even finding me jobs. This man employs me a lot. He's gotten me my job with CCFRP, with Surf Zone, and also dog sitting for him on the side. So thank you for all of that. To Tom, you have been a rock through this and really helped ground me in providing very useful and integratable feedback throughout this process, and in fact telling me when I'm doing too much, which is so very nice. I really appreciate that you have they're shaking their heads no over here. Yes, you do make me try to do more. It's nice to have the balance. So thank you, Tom, for encouraging me to have better boundaries and also provide me with and facilitate my process through all of this. And to Sherry, thank you so much. You started me on my remote sensing journey. I didn't know anything about it before that. And I know that you sometimes joke that I've surpassed where you are, but I wouldn't be here without you. And you've served as a confidant throughout this whole process, and I'm so eternally grateful for having your support throughout this entire thing. Now moving on to the very text-heavy slide. Okay, I have a lot of people to thank who have directly been involved in this process, so I'm going to try to go through it very quickly. To everybody at CSUMB and in the Ocean Predator Ecology Lab, thank you to my 2 undergraduates who helped me throughout my process. You kept me sane, and also helped facilitate me moving forward in this process. To Lacey, who met with me weekly, even though you didn't have to, very voluntarily throughout this process, to either brainstorm or to help me run through code. Very much appreciated. To Sarah, thank you so much for running my validations. Whether it meant me sitting with you or you sitting with me, I was always happy to have your input on how things were going and to hopefully have taught you something and you've given me something in return. To my lab mates, to Duncan, Bree, Kate, Dylan, to Eric, our volunteer boat captain, tagger, drone pilot extraordinaire, thank you so much for supporting me through brainstorming and through data collection and going out and actually doing the things and helping execute them. And special thanks to Ryan, who isn't here today but may be tuning in, who helped bounce ideas off of me and, similar to Tom, kept me grounded, and always what he left me with was, it's just your master's, it's not the end of the world, it's okay. So thank you for that. And to James, he provided me some funding when we were first starting up, so I really wanted to acknowledge him in taking a chance on me when Sal and I were both finding our footing. Now getting into all of the Moss Landing people. For data processing and analysis, thank you to Mike for helping me when I was lost in all of the millions of satellite data points, and my tiny little rinky-dink service could not handle it with R. Having your expertise and just your support was really helpful in facilitating the process and making it go a lot faster, because I simply did not know what I was doing, and you very, very much do, so thank you for that help. To Nate, who spent so many hours in his office with me going over whether it was just statistical theory or the application of these GAMs or even some of the visuals. Your input was especially helpful throughout this process, as I did not know how to do a majority of these statistics before coming in. So thank you for that. And then I also wanted to acknowledge Madi for the seasonal anomalies that I did. I used your setup of the different seasons, so having that handed to me via recommendation through Tom was really helpful. And then for the mooring construction, thank you to Patrick, Jake, Madison, Rick, and Karen, whether it was just talking conceptually with you or the acquisition of different assets. It was really helpful having you in there when those acoustic moorings were my babies. I really tried my best to make them as secure as possible, because I was really passionate about specifically this portion of my research, so you really helped with getting me those skills to get them out there. And in talking about getting them out there, how could I do it without the diving and without the boating and mooring deployment? So for diving, thank you so much to Diana Steller, to Jess, Logan, Bella, Noah, Roxy, and Kirsten. You were willing to brainstorm with me, to execute these things, and to, most importantly, really trust me under the water when we couldn't see more than a foot from our face. And I don't know if for those of you who aren't divers know, but that's so scary. But all of you really, really helped me, and I'm so grateful to have had you in my corner for all of the diving that I did. And similarly for the boating and mooring deployment, thank you to JD for helping me in talking all things safety, all things deployment, making sure that things were being executed properly, even when the boat broke down because Duncan brought a banana on the boat. Thank you for getting it up and running to make sure that we were able to get there and actually deploy them. You've also been a huge support, so thank you for that. And to Jenny and Helena for captaining for me, and to Jonah and Bruno for other deployment things, for being out on the boat with me and getting things prepped. To all of the graduate and IT support, thank you to Tara, who is on vacation right now, but you have been instrumental to not only me, but the entire Moss Landing community graduate students. I don't know if I would have been able to progress as fast as I did without your expertise in knowing when the timeline was, because again, I'm the first for our lab. To Katie, thank you for all of your library expertise in helping to find me resources that I needed throughout this process. To Misha and Silvestre for all of your IT support. To Petra for connecting me with funding for the boating that I did through Moss Landing, and to the MLMO greater community staff, faculty, and other students, thank you so much. And specifically to facilities, thank you to Matt, James, and Jose, who keep this place running, which is really, really special, so thank you for that. And then I also wanted to acknowledge my permits and parks, CDFW, the California State Parks, and Monterey Bay National Marine Sanctuary. Without your help, I wouldn't have been able to do my study in such a place. And also wanted to acknowledge my funding sources of the Coast Field Experiences Award, the Myers Grant, the MLMO Wave Awards, the Ryan Graduate Award, the Parker donation that helped fund the boating, and also Sal's Award, which I don't have listed here, which is the Otter and Shark Ecology Award. Now we get to the fun part. Okay. Have fun playing Where's Waldo. I have so many people to thank. Friends, thank you so much. I'm lucky enough to have a really huge support net of all my friends from all the way back in elementary school I've kept in contact with, who have supported me through this, through high school to college, and now here. You've all really uplifted me and made sure that I didn't spiral, and provided me with moments of levity and joy throughout this process. So thank you so much for making my time here enjoyable. And then I also wanted to thank my family, of whom there are some here today. You have also provided me respite and enjoyment in other ways, and you have been a constant in my life, thank goodness, throughout this entire process. Even when you questioned when I was on my computer during breaks doing my work, you still kept on with me. So thank you so much for believing in the fact that I did need to do those things at that point in time, and for supporting me whenever I asked for help or that I needed just simple things. Thank you for all of that. And in addition, there are a couple people who could not be here today, and the first of whom is my grandfather, who passed away during my second semester here, my first year. He was a great man. He was quiet, but he knew what he wanted, and we connected very early on. You can see from that image in the center where we would talk a little bit, have some similar interests, and then just be in each other's company. And he was always excited. I would call him on about a weekly basis and go out to visit him towards the end of my undergraduate and the beginning of my graduate time. And he was always excited to hear about marine science and hear what I was doing next. And so I know he would be really proud to see where I am now. And especially, I wanted to talk about my mom. My mom passed away after I graduated from undergrad, before I came here. She was my biggest cheerleader, always. I went into undergrad not knowing what I wanted to do. I was undeclared. She wasn't worried about me. Decided I wanted to go into science? Sure, go for it. Want to get a minor in Spanish? Yeah, sure. Want to be involved in sports and music? Yeah, do whatever you want. I'll be here, and I'll be on the boosters. Why not? And she was so excited for me. She actually got me into specifically marine science through finding me an opportunity to go to Florida and do shark and ray research there. So she was the catalyst for me and a huge support throughout all of my life. And I miss her every day, and I'm sure she would be so, so proud to see where not only I am, but where my sister is too, as she's in her grad school experience now. And so with that, thank you to everybody here, and I will take any questions at this point.
Kelsey, and that was a moving set of acknowledgments as well. So, all right, this is a time where we can take some questions from the audience, and the mic doesn't feed this room, but it's for the benefit of the people who are on Zoom. So I'm going to walk around, and then maybe I'll sit down. We can pass the mic around for anybody who has questions. So fire away. They can be softballs or a little challenging, or just clarifications. So anything's welcome.
Great job, Kelsey.
Thanks, Kevin.
So you talked a lot about anomalies, especially with temperature.
Yep.
And you had a pretty long data set,
Yep.
quite a few years. So my question is about an anomaly you didn't really mention except for in your intro, which is the El Nino Southern Oscillation.
Yeah.
So what effect do you think that would have on juvenile white sharks and their ranges based on whether it's a El Nino or La Nina, both in Monterey Bay and on a greater spatial scale along the coast, especially with regards to both surface temperature and turbidity due to different amounts of rainfall between the two?
Yeah, so good question. I think we didn't use that as a metric of one of the anomalies because it does kind of shift in the 3 to 7 year cycle, but we did kind of try to get at it by using the monthly and seasonal anomalies there. So in combination with that and the spatial, whether the models included all of that, we could kind of encapsulate that and then use that as a projection onto the characteristics of an El Nino or a La Nina. So knowing that El Nino years are much warmer would probably be manifested in the form of either a seasonal anomaly or a monthly anomaly or even any of the other climatological anomalies that were there, and then also in the spatial. So I think We would definitely, depending on what time of the year the sampling is done, we would see a different effect. So if surveys were conducted during the winter, we would probably see an increase as the waters would be warmer than typical. But then in the spring and summer months, we probably would see fewer as they're not spatially limited and can go elsewhere. And then the opposite in a La Nina year, where those trends would be contradictory. Does that answer your question fully? Sweet.
Thank you for your talk, Kelsey.
Of course.
Our boat conversation the other day, you mentioned that the tags were fairly long-lived.
Yes.
So will those tags, as the sharks develop and begin to roam greater and greater ranges, will they be able to be picked up on other receiver networks?
Yes, absolutely. So these tags that were deployed, if they were deployed well, can last anywhere up to about like 7 years long-ish. And so these tags are developed to communicate with receivers. So if there's a bunch of receivers deployed along California through a bunch of different institutions that we collaborate with as well. So if they grow and go further north, and they are near one of those receivers that has a compatible model to it, we will get those detections, and then whoever downloads those data will go, I don't know this tag, is this tag yours? And then they can send those data to us. So we can see where they're going as long as the receivers are out there to receive the transmission from those tags.
Has anyone picked up any of your sharks anywhere else?
Yes, Barb's network did, I believe, down in the southern portion of the bay, but not as much as the ones that we deployed in Soka Cove. Yeah, a couple up in Anyo. Yeah.
That was a great talk, Kelsey. Congratulations. So, not all migratory populations like this, where the animals discover a new spot, are permanent. Do you think that if we have a couple of cold years, would the sharks forget about the bay and would we stop seeing juveniles, or would they come back afterwards?
Good question. I think they're so highly mobile that they may just come up and see that it's too cold and leave. There have been a couple of recent cold years where we still do see them, just not as much as in the warmer years. So it's very possible that there could be a few that are like, hey, it's cold, but nobody else is here, I'm gonna stick around. Or they might come up, see it's too cold, go right back, and then still have the ability to go in between when it gets warmer. Yes, absolutely. Let me go back.
Thank you for explaining it. I understand it better now.
Yeah.
Appreciate that. Okay, so I'm looking at this high. That's
Yes.
if you look at this, the turbidity, but in the slightly cooler waters. And I get that. I understand that. But why would you have this mode where it's low above the heat line, where you'd think that, well, can you posit a hypothesis as to why there might be a lull there, why that high that you see in turbidity doesn't just extend all the way up to into the other warmer spatial anomalies?
Yeah,
Sea
so
surface
that
temperature.
light blue right above
why-
the red blob there. Yeah, that is a little bit trickier because this is it's a strange interaction between the two. I did talk about the Goldilocks of like when turbidity is high or when it's low, but the partial effect here is not as negative as when waters are extremely turbid. So it could just be that when the waters were warmer than the rest of the bay, slightly, not way more, and there was that intermediate zone of turbidity, that there was just the ability for the juveniles to not be locally restricted. So it could just be an artifact of those happening at the same time rather than specifically that being a distinct presence, because it is more towards the middle of that partial effect range than if it were a really, really dark blue there, where you can see like they are selecting for these areas when it's below the spatial anomaly and when it's above. So I think it has much more to do with the sea surface temperature spatial anomaly than it does have to do with the turbidity, if that makes sense.
Thank you.
Online, there's questions.
All right.
Nice.
Amazing, Kelsey. You are so smart. Proud of you. Okay, where's the question here?
Here's the question.
Okay.
In your opinion, what was the most exciting, interesting, fun fact thing you discovered in your results and research?
I think the most interesting thing that popped up was because I ran all of the models for the tagging portion and the remote sensing portion in advance, and then after I had already had a draft of my written thesis, Sal was like, but what if time of day has an effect? And I was like, okay, I will work on that. And then went back and ran those models, and seeing the really distinct trend of them showing up during non-daylight hours was like, okay, this makes sense now that we're seeing the opposite effect in temperature, because something else is happening when it's colder here and they're spending more time here. So I think that was the most like, aha, these things make sense here moment.
Okay, related to that, Greg Kaye is in the audience. Hi, Greg, and has weighed in and has a comment about the thermoregulation and wants to, if I phrase this as a question, talk about how the water temperature that influences the thermoregulation, but it's also the activity of the shark.
Yes.
So with regards to the surface swimming and underwater at depth behavior, how would you address that?
So I kind of alluded to it a little bit, where the juveniles have much smaller mass, so they don't have as great thermal inertia, or in other words, they can't hold on to their heat as well when they're surrounded by cold waters. So that's where the thermal regulation in the warm water comes from, and there was supposed to be a whole other set of research that I was going to do with a custom biologger, which I didn't get to because it was just too much, that would look into these trends where we would expect that if they are thermoregulating and seeking out warmer waters, that they would be exhibiting a slower, more turning swimming pattern towards the surface of the water. But then when they were trying to move through cold water, you would see them moving very quickly in straight lines to try and find the most suitable water. And I think the same would follow for if they were if they were exhibiting foraging behavior, where that would be a lot more rapid and quick, where they're expending a lot of energy, and that in moving that they're generating a little bit of body heat, but losing it to the environment before coming up and spending a lot more time, a little less active towards the surface to rewarm.
Okay, and Nate Spindle has a very challenging quantitative question.
Perfect. Hit me with it.
Are you ready?
Yes.
Did you name any of your tagged sharks?
Perfect.
Good one, Nate.
I don't think we named any outright. There was one that already had a name that we tagged. The largest shark of those that we tagged, which was at least 4 feet longer than any of the rest of them, who was like on the verge of potentially subadult, maybe shouldn't have been in the area but was still exhibiting the juvenile behavior, was named Emily. So that was a name that I didn't come up with. It was already referred to as Emily beforehand, but all the others we did not name. So.
All right. Any other questions in the room before we take her back and further cross-examine? All right, well, let's give Kelsey another round of applause. Thank you, everyone online.